high-throughput injection with microfluidics. Using Picoinjectors is a groundbreaking approach that allows precise and efficient addition of reagents into microdroplets at extremely high speeds. But why is this important and how does it work? Imagine needing to add tiny amounts of different chemicals into thousands of microscopic droplets for drug testing or genetic analysis. How could this be done quickly and accurately? Traditional methods struggle with precision and speed, but picoinjectors offer a solution by using electric fields to introduce minuscule volumes of liquids into individual droplets. This means scientists can perform highly complex experiments, such as drug screening, biochemical reactions, or genetic sequencing, at speeds of up to 1,000 droplets per second. The ability to control injection volumes down to sub-picoliter levels ensures that each droplet receives the exact amount needed, preventing waste and improving accuracy. Think about single-cell studies where different cells need to be exposed to specific molecules. This technology allows researchers to fine-tune treatments at an incredibly small scale. Another example is in personalized medicine, where scientists could test multiple drug formulations simultaneously within a single microfluidic chip, significantly speeding up drug discovery. But how does the Picoinjector work? It uses a pressurized channel that releases a reagent into a droplet when triggered by an electric field. The electrode momentarily disturbs the surface tension between the droplet and the reagent, allowing the two to merge without causing unwanted mixing with other droplets. By carefully adjusting the pressure, voltage, and droplet speed, precise amounts of reagents can be injected selectively, enabling combinatorial and serial additions of different substances. A practical example of this technology in action is high-throughput screening for new pharmaceuticals. Rather than testing one drug candidate at a time, researchers can rapidly test thousands of variations in parallel. Another exciting application is in synthetic biology, where engineered microdroplets serve as mini laboratories to study biochemical pathways and design novel biological systems. What are the challenges? Ensuring that injected droplets remain stable, preventing contamination, and optimizing the system for industrial-scale applications are all areas of ongoing research. But as microfluidic technology advances, picoinjectors could revolutionize fields ranging from medicine to materials science, bringing us closer to a future where diagnostics, drug testing, and chemical synthesis occur at speeds and scales never before possible. Could this technology one day lead to automated on-demand medical testing or even micro-robotic drug delivery systems inside the human body? The potential is immense, and continued research in microfluidics is pushing these possibilities toward reality. Picoinjection is a high-precision technique in microfluidics that enables the controlled addition of reagents into individual droplets at extremely high speeds. It utilizes electro-microfluidic principles to achieve sub-picoliter precision in reagent delivery. The process involves a pressurized microfluidic channel containing the reagent, with electrodes applying an electric field to disrupt the interface between the droplet and reagent channel, allowing precise fluid transfer. This technique is particularly advantageous for high-throughput biochemical assays, drug screening, and genetic sequencing, where small volume injections ensure accuracy and reproducibility. In genetic analysis, picoinjectors are used to introduce sequencing enzymes into single-cell droplets, allowing massive parallel processing of DNA fragments. Picoinjection functions like an inkjet printer, where droplets are precisely injected with reagents just as an inkjet deposits controlled amounts of ink onto paper. Picoinjection operates by utilizing the principles of electrocoalescence, where an applied electric field destabilizes the oil-water interface of the droplet, enabling the transfer of reagents. The process relies on the balance between Laplace pressure, surface tension, and the applied electric force. The reagent remains confined until the electric field is activated, at which point the interface ruptures, allowing controlled injection. The droplets maintain their structural integrity due to stabilizing surfactants, preventing unwanted merging even when in close proximity. This level of control ensures highly accurate reagent additions in dynamic microfluidic environments. In single-cell proteomics, Picoinjection enables the sequential addition of lysis buffers and fluorescent markers into isolated cell droplets. Without cross-contamination, the process is akin to filling tiny balloons with exact amounts of gas, where each balloon expands only when triggered by an external force. One of the key advantages of picoinjection is its ability to operate at kilohertz speeds, making it suitable for large-scale biochemical reactions. The injected volume is finely controlled by adjusting injection pressure, electric field strength, and droplet velocity. By increasing the reagent channel pressure, injection volume is enhanced, while raising the flow rate of the carrier fluid reduces injection time, leading to smaller reagent volumes. The ability to switch injection on and off at high frequencies allows for complex reagent delivery patterns, optimizing serial and combinatorial chemical reactions in microfluidic systems. In enzyme kinetic studies, picoinjectors deliver precise volumes of substrate and inhibitor solutions into reaction droplets at high speed, enabling real-time observation of enzymatic activity. It functions like an automatic espresso machine, dispensing exact amounts of liquid at high speeds with fine-tuned control. Picoinjectors allow multiple reagent additions in sequence, enabling complex biochemical assays within droplets. Serial injections introduce different reagents one after another, while combinatorial injections permit the simultaneous delivery of multiple compounds. This approach is particularly valuable for applications such as directed evolution, where genetic mutations are tested in parallel, and for high-throughput drug screening, where various drug combinations are evaluated within microdroplets. In pharmaceutical research, picoinjectors introduce different drug candidates into droplets containing tumor cells, allowing rapid screening of therapeutic effects. It is similar to a robotic bartender precisely mixing different ingredients in a drink, ensuring each glass gets the correct combination. Picoinjectors are designed to be compact and modular, allowing integration with other microfluidic components such as droplet sorters, cell encapsulation units, and reaction chambers. By combining picoinjectors with microfluidic flow cytometry, real-time fluorescence-based analysis of reactions is possible. Additionally, microfluidic sorting can selectively direct injected droplets toward different downstream processes, such as further analysis or storage. In synthetic biology, picoinjectors are integrated with microfluidic droplet sorters to screen engineered bacterial strains producing specific metabolites. This setup is like an assembly line where different machines handle separate tasks but function seamlessly to produce a final product. Picoinjection plays a crucial role in single-cell studies, where precise reagent delivery is necessary for single-cell RNA sequencing, single-cell metabolomics, and proteomics. By injecting lysis buffers and reaction reagents into individual cell-containing droplets, picoinjectors allow the extraction and amplification of cellular content at an unprecedented scale. This approach enables high-throughput analysis of gene expression, protein function, and metabolic activity in heterogeneous cell populations. In neuroscience research, picoinjectors are used to introduce fluorescent calcium indicators into neuronal cells encapsulated in droplets, facilitating the study of synaptic activity. The process is similar to a chef adding different spices to individual meal portions to create unique flavor profiles. Despite its advantages, picoinjection faces challenges such as droplet size variation, reagent backflow, and precise synchronization with droplet formation. To optimize performance, researchers use surfactants to stabilize droplet interfaces, fine-tune electric field parameters to prevent unwanted coalescence, and employ high-speed imaging for real-time monitoring. Advances in microfluidic design, such as improved electrode configurations and feedback-controlled injection, enhance precision and reproducibility in picoinjection applications. In precision medicine, optimized picoinjectors are used to deliver custom drug formulations into patient drug organoids for tailored treatment testing. It is similar to a high-performance car engine that requires precise fuel injection timing for optimal performance. Future advancements in picoinjection technology will focus on increasing automation, miniaturization, and compatibility with lab-on-a-chip platforms. AI-driven real-time control of injection parameters will enhance reproducibility, while novel electrode materials will reduce energy consumption and improve injection efficiency. Additionally, integration with biosensors will enable real-time monitoring of biochemical reactions within droplets, opening new possibilities for diagnostics and drug development. In personalized cancer treatment, AI-optimized picoinjection systems could test hundreds of drug combinations on patient deconditioned tumor cells to identify the most effective therapy. Picoinjection technology is evolving like self-driving cars, becoming increasingly autonomous and precise in decision-making. As picoinjection-based microfluidics moves toward clinical applications, regulatory approval and ethical considerations must be addressed. Ensuring sterility, biocompatibility, and reproducibility in medical applications is crucial for patient safety. Additionally, standardization of microfluidic devices will be necessary to ensure widespread adoption in pharmaceutical and diagnostic industries. Ethical concerns related to personalized medicine, such as data privacy and accessibility, will also require careful regulation. Regulatory bodies are developing guidelines to ensure that picoinjection-based microfluidic devices used in clinical diagnostics meet stringent safety and efficacy standards. Navigating the regulatory landscape for picoinjection is like obtaining flight clearance for a new aircraft. Extensive testing and safety checks are required before widespread deployment.